and welcome to another episode of Awakening Your Health Potential. And today I have the very interesting and clever Margaret Smith, who has a Bachelor of Science and did her honours in Bachelor of Science and then did her PhD in molecular genetics. And I've recently come to know Margie through her business, which is Margaret, you know what? I think you're going to explain this better than me. So okay. Let me introduce you and we'll start off with maybe a little bit about SmartDNA and then how you got into it.
Okay. Well, look, thank you. Thank you for the intro. And yes, I work at SmartDNA and I am a molecular geneticist, which means I have this fascination with DNA, whether it's from human beings or animals or plants, because, we're all connected. And DNA is certainly a really interesting piece to help us understand our health, for sure. So I look forward to talking to you today about all of that.
So how did you get interested in genetics?
Well, if we take a step back, I think I was kind of born a scientist because I've always been really interested in, the world that we can see and then the world that we can't see, kind of what's hidden and how do we explore those hidden parts of nature and science. And so I was always curious as a kid. And then in my family, I had a sister, Jennifer, and she was born with a neural tube defect. So she was born with spina bifida. And that really had a profound effect on me because I was really challenged as to why, I would have a sibling that was born with this birth defect and being really puzzled by it. And with my curious nature, I confronted my parents one evening and suggested that there might be something wrong with their DNA.
How old were you?
I think I was about 8 or 9, and that didn't really go down particularly well. But, I was kind of halfway right because we've come to understand that, neural tube defects, spina bifida is in part DNA and risk, and then also that intersection with nutrition. So it turned out, who knew, B vitamins and changes to that what's now the methylation pathway can contribute to neural tube defects. So I wasn't popular as a child at that point, but I think my parents came to understand that they had produced this little being with an inquiring mind who may not always have the best choice of words, but was, always interested in the world around her. So, and then, I sort of moved into working in a hospital situation. I worked in anatomical pathology, so I saw these weird and wondrous tissues that human beings could grow and were often cut out of their bodies and ended up in pathology. So that was certainly an experience. And then I really became more interested in probably more the academic side of science. And I was invited to Melbourne Uni for an honours year. So I had a scholarship. And following on from that scholarship, I worked on Duchenne muscular dystrophy, so another genetic kind of disease, but I looked at proteins and I looked at the muscle tissue from those kiddies. And then somehow or other, I managed to get a BSc honours, which meant that I could progress to doing a PhD, and was lucky enough to get a scholarship of, I think it was around $14,000. So the rest of my funds were made up from working in private pathology every weekend. So I think that just goes to tell the tale of when you're really passionate about something, it doesn't really matter how many hours you work because you just love what you're doing. And I used to feel incredibly proud of going to Melbourne Uni and working on my PhD, which was on early onset Alzheimer's. And that definitely had a genetic basis because some genes had been discovered. And there were about 8 to 12 families in Australia that had early onset disease, and they had no known genetic cause. So I found myself in front of a -70 freezer one morning, and I was very excited because I'd planned my experiment. I knew what I was going to do, the genes we were going to look at. And I was about to lift the lid, and Professor Colin Masters, who had given me this wonderful opportunity, said, "Margie, what are you doing?" And I said, I'm really excited. I'm about to take some tissue from these half hemisphere frozen pieces of brain tissue, and we're going to look for those mutations now." And he said, "Oh, great." He said, "Just remember one thing." I said, "Oh, what's that? What's that?" And he said, "Just remember that the people who donated their half hemisphere of frozen brain tissue for you to study have got living family members, and they're going to want to know what you find." So it was very leveling, because it took me back to my mainstream medicine, to everything has to pan out, all the numbers have to match, everything has to be 100% accurate. So it really was a really salient point that whenever people donate tissues or blood samples, there's probably other people who are going to want to understand what those findings are. So that did put a serious tone to the work that I was doing. And we did find mutations. And I think the most important aspect was, yes, it was great to find mutations, but as a scientist, it gave me a far greater understanding of human beings. Mm. And it's all good and well to be in a laboratory and have a blood sample or a tissue sample and analyse it. But when you understand the impact that information has on a family, and you understand about coping mechanisms from family members, and especially if there's their partner who's got early onset Alzheimer's, often there's that feeling of helplessness and what can they actually do to contribute. And in one of the families, it's really fascinating, the husband actually took up genealogy and he mapped out the whole family tree right back to the UK when the original founder members came to Australia. And coincidentally, at that same time, I'd received a sample from up in Sydney, and it had this family had the exact same mutation that I'd found in a family in Melbourne. So the geneticist that was part of the team that I was working with connected those families and with this other family member who.
Wow. So they were related.
They were related, yeah.
Wow.
Yeah, back in time. And so they'd basically just moved along the east coast of Australia. So, yeah, it's really, really fascinating. Genetics can connect people that have been lost to each other for a long period of time, even when you're looking at disease processes. So I learned a lot more about people and feelings and communication than I ever would have without that opportunity to work on my PhD. So nothing ever happens, by yourself. It's always with a group and a team of people. So there were lots of other specialists involved, amazing neurologists who can put together small amounts of information into a wonderful story. Pathologists. I mean, we learned so much from that work. I think one of the original thoughts was, if we look at early onset Alzheimer's, will that help us to understand late onset Alzheimer's? And it turns out the pathologies are quite different.
Right.
Different underlying genetic mechanisms at play. Even though the disease is sped up and some of the pathologies are the same, it didn't really help us understand the pathways that are involved. It helped us to understand how those genes, what happens to those genes when they have a mutation in them, the proteins. But yeah, you can't really compare early onset disease with late onset disease. I mean, late onset disease, I think we have some determination over through diet, lifestyle, and our understanding our genetics. So nutrition's a key part to all of that. But with early onset disease, they're quite major genes that have, fundamental flaws in those proteins.
Yeah, right. Well, that's one of the things that I really love about the SmartDNA testing that you do is all the nutritional things and exercise and weight management. I've noticed with some of the reports that I've been through recently, it's like the obesity profile fits in with a whole heap of the other profiles, and for one person I've looked at recently, he's got a massive cardiovascular risk. Like, it's come up in lots and lots of different areas, but because he's always managed his weight, none of them have actually transpired.
Right.
So it's really, and he's in his, I'd say late 70s.
Yeah. Okay.
So, yeah, it's quite interesting. And then whereas other ones I can see with people where their weight's gone up suddenly, like these other things are starting to become an issue. And I've also noticed that with hormones, you know, where women are, particularly whether they're going into that perimenopausal phase and postmenopausal phase, it kind of then activates a whole heap of other stuff if you're not taking care of it, which explains so much in terms of people's symptoms at those kind of ages that I hadn't actually put together. But it's kind of all mapped out in the SmartDNA genomic test, which I just find unbelievable. And it makes it so simple to work towards preventative health. So I guess I'm totally excited about this area. And what amazes me is that I actually work in this area of preventative health, but I had no idea it really existed until kind of, I guess, middle to late last year. And so how new is it? How new is this technology?
Well, we started looking at nutritional genomics in 2009, and that was really like super leading edge information at that point in time. And since 2009 up until now, I mean, we have gleaned more information about a lot of those genes. There's more research, more studies being done.
Yeah, it's like constant research, I would imagine. Like, it's
a
big developing area.
Yeah, it is. And the cardiovascular area that we look at, I mean, I don't know of any other way that you can know the information around if you're high cardiovascular risk, what your nutritional intake should be versus someone who's more of a neutral risk profile. And it's all based around a Mediterranean style diet. And that's one of the most popular diets, right? The most studied diet on the planet. But when it comes to cardiovascular health, there are tweaks to the amounts of fats, proteins, and carbs that need to be intaken based on your genetics. So, you know, this is one.
Even the concept of how exercise fits into it. You know, it's we have all these like general. My undergraduate degree was in exercise science, and we have all these like real great information on how to keep someone healthy and what they should be doing. But then it's like the reality is there's just not a way of saying this is how everyone should exercise, this is how everyone should eat, that actually works for every single person.
Well, I think even too, like in that high cardiovascular risk group, we talk about a little bit more aerobic exercise. And the science behind that is that we can actually grow our brains via exercise, right? We actually grow BDNF, brain-derived neurotrophic factor, and that's all involved in memory, and that's in our frontal cortex. So, you know, that high cardiovascular risk group does have associated risks with declining memory during aging and it being sort of brought forward maybe into their mid-70s. That's not always the case because, let's be clear, when it comes to cognitive decline or, you know, memory loss as we age, there are many, many, many genes involved in that, not just one gene.
Yeah.
If there was one single gene, mainstream medicine pathology would use that, but there isn't. So the gene we look at is fantastic for cardiovascular health and nutritional genomics, but it's not an accurate marker for cognitive decline. So it's impossible to say that, yes, this gene's associated with this and you're definitely going to develop that disease. That's just not the case. So I think
it gives you like when you do the tests, the 160 tests that you do with that genomic wellness test, it gives you such a good baseline of all the different things that you can do to help support those weaknesses that I call them like your health weaknesses. It's like you can find out what those weaknesses are that don't necessarily have to express. If you've inherited these genes, they don't necessarily express themselves, but they often express themselves, and you might be able to correct me in terms of scientifically, like with some sort of trauma, like trauma to the physical system, trauma to the biochemical system, or something happens where there's something, and then there's a time when things then change.
Yes, I mean, like a really simple one will be, let's just presuppose we're looking at the usage of B vitamins and say, based on your genetics, your enzymes operate a lot more slowly. So they're not as effective, and your need for vitamins may be more demanding in terms of what your requirements are. But you suddenly decide that you'll go on a vegetarian diet. So your intake of B vitamins now diminishes dramatically, right? So you have an increased need for B vitamins, yet your dietary intake is very low in B vitamins. So there's a collision course now between what you actually need in a biochemical sense, yet what you've chosen to eat for maybe reasons that you don't want harm to come to animals. You just feel that plant-based eating is going to be totally the thing for you to do. And so there are, many populations, even for religious reasons, will not eat meat. So those individuals can have chronically low levels of B vitamins, and then in terms of biochemical markers, they can have markedly elevated levels of homocysteine, which is not Homer Simpson's brother-in-law, right? It's actually homocysteine is a byproduct of a methylation pathway, right? And if you have adequate B vitamin intake, you basically snap apart homocysteine to methionine, and you make these lovely cysteines. And enzymes love cysteines, okay? That's, our body runs on these things. So you can see that, for whatever reason, people will make different nutritional choices, but it's really important to understand in a nutritional sense what your need for dietary vitamin intake may be, because it may be more than the daily recommended intake. So, they're the kinds of intersections where people will make a nutritional choice, but it may be at odds with what your actual demands are for B vitamin intake, for example.
Yeah, and Parkinson's is one that's come up for me recently. So, like we know that a lot of kind of head injuries can lead to Parkinson's later, but would they have to be genetically predisposed to Parkinson's? Because the ones that I've seen that have Parkinson's, for example, I've just had someone recently and he had a terrible accident, fell down the stairs, in his probably late 60s. And, I've gone through his genomic test recently, and there's, I think, 2 or 3 genetic markers that says he's predisposed to low dopamine. He's sensitive to specifically Parkinson's drugs. So it's like that it kind of blows me away. And then I've got another lady she's actually a young mum, but she's been possibly diagnosed with early signs of Parkinson's. And the symptoms have come out since the whole COVID restriction, when she was just so stressed trying to school 3 kids at home, still work, and these symptoms started coming up. So it's like this stress, the extra stress on the system kind of pushed these genes. I don't know if that's scientifically how.
Right, yeah, I think there's a whole bunch of points to what you've just made. I mean, so if we looked at Parkinson's disease as a familial heritable disease, we'd look at genes like PARKIN1 and PARKIN2, but we're probably not talking about that. We're probably talking about so-called sporadic cases, these just incidental cases in a family where someone will have, for example, as you said, Parkinson's disease, and they may be part of aging. It's not normal aging, but you're right. Then when we look at nutritional genomics, I mean, we know for people with Parkinson's disease, dopamine, low levels of dopamine is an issue. Low levels of glutathione is another issue for those individuals. So then you can start to look at the pathways and what support is necessary. It may not be that the genetic predispositions to low levels of dopamine is a trigger for developing Parkinson's. But certainly, you talked about head trauma as being, another area that is associated with development of Parkinson's disease. So we're not necessarily looking at all of the pathways involved, but from a nutritional sense, what we're looking at is what we can actually do to support those people with these neurodegenerative type diseases and disorders. And I think, nutritional supports are really very important in those instances.
Yeah.
For sure.
So have you done a whole heap of study in nutrition as well? How did that all come into it for you? And actually, what we haven't really defined for listeners is what nutritional genomics is. So maybe you could start with that.
That's right. We've launched straight in. So nutritional genomics is really understanding where nutrition meets your genome, your DNA. So we're looking more at personalisation for people. So we've talked about B vitamins, and I think one of the most known genes is MTHFR. Most people have heard about MTHFR and the need for B vitamins, and really that's just the starting point, because in that entire pathway, we're looking at other genes associated with a need for not only whether it's folate or a methylated folate, but vitamin B6, B12. And for some people, more need for vitamin B2 based on genetics. So no, I don't have qualifications in nutrition. I have done some studies in nutrition, but I don't have any qualifications. But as a geneticist, basically my area of interest is looking at genes and understanding, is their enzyme faster, slower, how is it affected, and how does that interact with nutrition. So there's a lot of keeping up to date with literature and studies, and also understanding where that fits with pathology. So for example, let's say your MTHFR enzyme is very sloppy, and this is the first place, or it's slow. This is the first place where we metabolize B vitamins, whether we get that, might get it from protein, for example. Or you might be supplementing because you feel better when you supplement. You may not know what your MTHFR enzyme is, but think about it, as an enzyme that's there to basically do one thing: add this little methyl group. Okay, it's the first step in the process. But if your enzyme is slow, it's going to take a long time to do that.
And I think of it like a cog. So it's like
a cog. So it's grinding away. It's just going really, really slowly. And other people might be whizzing around, as you've said, but this is going really, really slowly. So one thing that individuals may do is take a methylated folate. So we're saying to MTHFR, okay, you're not really doing the job as fast as we would like. We're going to step in behind you and add that methyl group to folate. So you can actually tick away through the rest of the pathway. Now, the thing about that is, if you're going to supplement and do that, if you're going to actively supplement every day, understand that the biochemical consequence of that is that you are going to push levels of homocysteine down. Now, homocysteine is a chemical that's associated with, if it's elevated, cardiovascular health, issues with cardiovascular health, and also brain function as well. So you don't want to have these elevated levels of homocysteine. Other reasons for elevated levels of homocysteine are people who drink too much alcohol, other medications, and primarily dietary choices that don't contain enough B vitamins. So, for example, in Asia, where I've done a lot of work, I've seen homocysteine readings of 30. Okay, now the normal range is between 8 to 10. Okay, 6 to 8 is really good DNA repair. If your homocysteine reading is between 6 and 8, that's fantastic. So just
for the listeners, so lower homocysteine is better because homocysteine interferes with you being able to repair your cells.
That's right. That's right. And it's a barometer. It's a marker for DNA repair. So between 6 and 8 is really fantastic DNA repair. But you don't want to be taking a methylating agent every day just because your MTHFR enzyme is a little bit faulty, because all you're going to do is drive homocysteine lower and lower over time. Remember, we want to keep it between 6 and 8. So it might be that you methylate every third day. May not be every day. And if you use homocysteine as a marker for what's actually happening in your pathway, that's a great way to keep track and make sure that you're not pushing homocysteine too low. If you push homocysteine too low, then you need to look at glutathione support to drive the pathway back in the other direction. So kind of think about it as, your DNA changes as being wonderful markers for what's wrong. But if you want to actually understand the biochemistry or pathology of what's going on, then, there are other tests that you can actually do to see what's happening in real time for a person. Mm. But I think it's really critical to understand in the first instance what your vulnerabilities are. And without that information, you wouldn't even know for cardiovascular disease what kind of diet to go on in terms of your macronutrients, right? So
totally. And even with the MTHFR gene test, which you can kind of get done by itself, and a lot of people I find do know about that. But it's like, when people say, oh, why does this nutrient show up that I need to, because I use a lot of muscle testing. Why is it always this, Yep. And having a look at their genetics, it then reveals like how and why. So it's like looking at those things that you're seeing that the body needs and it can't get enough out of its food through periods of time.
Right.
If you actually look deeper, it's like, that's actually why.
Exactly. And there are other parts to the pathway. I mean, we're looking at genes and proteins that are associated specifically in a nutritional sense around the need for B vitamins and methylation.
Which is
but if you looked at the entire pathway, there's hundreds and even thousands of other genes involved. But when you look at it in a nutritional sense, it's kind of a summary version of these are the key points that you need to know.
Yeah.
Absolutely.
So the other company that a lot of people have heard of is 23andMe.
Right.
What's the difference between the way you guys work? Actually, and even people, I don't even think people understand, even ancestry.
Right.
I mean, it's not nutritional at all, but maybe you can kind of define what the difference between those sort of, what people are looking for and what they get out of it.
Yeah, okay. So if we look at basically there's a fundamental difference in terms of technologies. So Ancestry and 23andMe use these very large arrays. So you get hundreds of thousands of pieces of information on your DNA, whereas our tests, we target specifically to nutritional genomics, and something that we can do something about in terms of, nutrition, your lifestyle, and exercise. So for the other companies, they may not have the SNPs that we look at. In fact, I know they don't. We look at some copy number variations. So did you get this gene from mum and dad or didn't you? So sometimes you don't inherit some genes from your parents. Yeah. So those glutathione enzymes, for example. So you won't get that information from 23andMe or Ancestry. And then with those large arrays, they're unvalidated. So the data may not necessarily be 100% accurate, whereas our test is validated. We've used something called genome in a bottle, and where the genotype is known, and when you run your assay, you know the genotypes that you should expect to see. So we've validated our test.
So that's why, some people, like someone I was talking to recently was saying, he's been told by doctors that they're a bit of a gimmick. And I'm like, definitely in terms of gimmick, I can say that SmartDNA is just, the information there is totally usable. to improve your health. That would fit in. If it's not validated, then
the arrays are not validated, so they're not exactly error-free.
And this
Ancestry Ancestry basically is looking at mutations that are found, or DNA changes that are found within countries. So they're called founder mutations. So if you harbour those founder mutations, that's how they can kind of work out whether you've got Irish ancestry or a bit of Scandinavian
Okay. So you don't get the detail in terms of how to use it for your health. It's more to do with like what your background is.
Yeah, it is. Although there is other information in there, it's just that with our testing, we've targeted specific genes and gene changes, which may not be on those arrays.
Yeah.
So, and with some of the early arrays, the information can be quite confusing because DNA has two strands, a forward and reverse strand. So it was never clear on those early arrays whether they targeted the forward strand or the reverse strand on the DNA. So that affected the interpretation. So basically what you end up with, data on an array you can upload to another party or company's program, and you can generate a report. But just remember, if there's an error made in the array data, that's going to be compounded in the report that you actually receive. So there's no real way to check that data. So there have been differences, and we have found that the array was actually the point of error.
Yeah, right.
So, I think using unvalidated information to provide a treatment plan for a patient is not really a very good way to go, in my opinion.
It's not relevant either.
No. So, you've got to have information that you can do something about to actually help a patient. And also in looking at a report, I always like to think about the good things in a person's report, areas where their health is not likely to be impacted, and then also the areas where some support is needed. Because prevention is everything. I mean, basically where I've come from in mainstream medicine, we don't really work in that area of prevention. We wait until there's a disease process. And then the way that disease process is looked at is very much often tissue specific. So if there's a problem with your kidneys, well, you're off to the kidney department, the renal ward. If it's heart, it's there. And so sometimes I think that we miss a lot because we don't really see the person in a holistic way. We don't bring everything together.
Yeah, so totally agree. It's and that's what I really love about this, because again, so many of those genetic markers that you're looking at have like a broad area of health that it would be affecting. So, not only are you looking just at the genetic marker, but also like looking at how that's going to impact the system and how you can support it to prevent all of those areas.
Yeah, if you just think about methylation, I mean, when you say that word, there's been now 100 million methylation things happen in my cells in my body just when I said that. Because it's happening all the time.
Yeah.
Like it's not like there's one thing that...
Just say that again.
Yeah, it's going on 100 million times in my cells right now as we speak. And so it's not something that we cognitively have control over, right? It's something that our body does for us. Yeah. So in knowing that information, we can support it. Because if there's one thing that we want to be faithful in our lives to us, it's our DNA, right? It may not happen in relationships, but you want your DNA to make faithful copies of itself.
Yeah, and the good ones and not express the not so great ones.
That's right. That's exactly right. We want that to happen. Yeah. So that's a really important piece to understand, that those faithful copies. And as we age, you've got to understand we've got little spell checkers that run around checking those A's, C's, T's, and G's on our DNA, and they run around and they make sure we haven't made a spelling mistake, because if you make a spelling mistake, you might make a protein that is not going to be helpful to you as you age. So you really want to support your body as you're aging in terms of nutrition, because that's certainly something we can control.
Mm. And then obviously things that also protect the cells are like reducing chemical exposure and heavy metals and
right
that thing.
Yeah, for sure. And I mentioned those GST enzymes just before, those glutathione enzymes, GSTT1 and GSTM1. Now, you either get a copy of those from mum and dad or you don't. And like, they are major detoxification enzymes. They're kind of like magnets for any breakdown products, from pharmaceutical drugs through to chemical exposures. I mean, even just in kitchens at home, there's plenty of chemicals that sit around there that we run our hands through, we breathe in.
Yeah.
So if you don't have those glutathione enzymes, then when you set up detox, a detox is happening by the liver. You don't have that capacity, so you have to use this non-enzymatic pathway, which is really slow. Even think about mercury exposure. I know that this is a little bit contentious, but if you've got amalgam fillings and you don't have those glutathione enzymes, then there have been studies showing that, you can end up with mercury toxicity.
Yeah.
So, next time you get a filling, my advice would be try and not get, mercury amalgams. I know that
as well. It's so unnecessary.
Yeah. Mm. And even children on the spectrum, one of the things they found was that that often those glutathione enzymes are deleted; they're not present, so their ability to detoxify is reduced as well.
Mm.
So there's a whole lot of biomedical evidence for nutritional understanding as well for those kiddies.
Yeah, absolutely.
But yeah, like, detox is a major area in the report, and as I say, we are one of the few companies that actually look at the copy number variations of those glutathione enzymes. And just to give you some idea, 50% of the Caucasian population has GSTM1 deleted, and around 80 to 90% of the Asian population has GSTT1 deleted. So those two enzymes. So basically, when you have them deleted, you don't detox as well. So
it's glutathione. It's the one related to glutathione.
Glutathione-related enzymes. So that's when you need to look at nutritional support. I mean, vegetables that contain sulforaphane, your broccoli, cruciferous vegetables, 3 to 4 times a week is hugely important when you don't have those enzymes present.
Yeah.
And just out of interest, Japan is a country where large percentage of the population have both of those enzymes deleted. So, and Australia is increased risk for GSTM1 being deleted as part of the Caucasian population, even though we're a bit of a melting pot now. But, when you start looking at those enzymes, the other issue around them is if you've deleted in those enzymes, then your need for vitamin C can be increased above the daily recommended intake. And they also found in people that have both those enzymes deleted, increased cancer risks. So we know vitamin C is anti-cancer, right? So, please make sure when you have those enzymes deleted that you're at least meeting your daily recommended intake for vitamin C. So, we're not, everyone looks at, like, for example, the enzymes for vitamin A and, eat your carrots. But, we're actually talking about beta-carotene. We're actually talking about the bioactive chemical components in these foods that interact with our DNA. Omega-3s can basically reduce inflammation in one of the one of the inflammatory markers called TNF-alpha. It actually directly interacts with the driving part of that gene, the part that controls that gene, and can dial down inflammation. So,
it's like so specific. It's like if you know where that SNP is, you can just come in and support it.
Yeah. So it's, people go, it's food, but we're actually driving down to the bioactive parts of food and how that interacts with your DNA.
Yeah. And then I guess it just enhances that old saying of you are what you eat.
Yeah, and I think now with our gut microbiome, not only are we what we eat, but we're also what we absorb through our other genome, those bacteria that are sitting there as well. You know?
Yeah, absolutely.
So, we're now going to start seeing this burgeoning of connection between our gut microbiome and our innate DNA.
Wow.
It's kind of like, who's in charge of us? Is it us and what we eat, or is it that bacteria sitting in our gut, that are controlling us?
Yeah. Yeah, it's like when I often get, kids in and mum's going, oh, their behaviour just gone off the chart, and I find a parasite, and I'm like, is it like your child's been taken over? They've got a parasite, it's like they get taken over. Yeah. And you can see, and then their behaviour just drops straight away. So, yeah, it's quite fascinating the way the gut influences. So,
there's so many axes from the gut to our lungs, and even in this time of COVID, just our, lung capacity, and then also what's happening in our gut microbiome, because that's where our immune system sits.
Yeah.
So, there's the gut-brain axis. There's the gut-skin axis. I think if we can just come to terms with and accept that everything is connected.
Yes, absolutely.
Start with ourselves.
Yeah.
Think about that and what's happening in our bodies, and then think about what we think, not only what we eat, but what we think, what we feel, our interactions, and just look to nature as well. Absolutely. Everything is connected, 100%. Everything is connected.
Yeah, absolutely. That's a really beautiful way to round off. But I do want to ask you where, what your highest kind of vision for this sort of knowledge for people would be, like where you see it going.
When from the moment we're born, we can have this information about ourselves. Like fundamentally, that would be great. When there's a newborn, we look for inherited disorders of metabolism. There's a few of those that look for when children are born. But if we looked at nutritional genomics in terms of what's a path for this individual for the rest of their lives in terms of nutrition and preventative health, that it would be there, that we would include the gut microbiome in that data set as well, and map and follow individuals. Because remember, they inherit, you inherit your gut microbiome from your mum. So, and then there's antibiotics and so on. There's other influences on our gut microbiome. But just remember, 50% of what we eat determines what our microbiome looks like. So for the future, I'd like to see this just be available for everybody to have access to.
Yeah,
absolutely.
So
that, everyone gets an opportunity to age in a healthy way and be healthier for longer. It's one thing wanting to have a longer life expectancy, but it needs to be a healthy one. And I think that's definitely where we need to head.
Yeah. Wonderful. Can you let people know where they can find you and about your book as well? I've got for those watching on YouTube, I've got a copy of the book right here.
Oh, right. So I wrote a book called Gene Genius. So you basically get to be the genius and understand about your DNA, and there's a whole lot of fun things in there. So you can remember the names of genes. Kind of
case studies and, like easy to put into context for people as well. Yeah. Yeah.
And you can find us at www.smartdna.com.au and look at our website, and that's where you can certainly buy a copy of the book as well. So
yeah. And do you have a list of practitioners that
we do? If someone wants a test and they live in a particular state or region, that we will try and find a practitioner for them for sure.
Yeah,
absolutely.
Yeah, great. And it's so easy, just so I can, it's so easy. They send out the little testing kit in the mail, you send it back, and it gets looked at, and it's really, really, really simple. It's fantastic.
It sure is. It sure is. And it's a great way to get a roadmap for health moving forward, for sure.
Yeah, and proper long-term preventative health, like on another level. So, yeah, I think it's amazing. I think that, more practitioners should know about this, that this is available, especially people looking at kind of long-term health And yeah, I reckon making it available. I've kind of done all my family, and I'm now looking to do like that, like my mum, my dad, and myself, my partner, and now I'm looking to do the kids. So it's like if I can get that information with I've got 2, 5, 6, 17, 18. The 17, 18-year-olds, I think they'll be the last one to get done, because I feel like they're probably in the phase of life where they don't really care anyway.
Yeah, right.
And I don't have the control anymore. So if they want to do it, I'll do it with them. But I feel like the little ones, I'm still in control of their nutrition and how to support them. So I think it's super important too. So,
well, let's hope that everyone's compatible in the kitchen, right?
Yeah, well, it can't be too different if, we're kind of mostly connected.
Mostly connected, that's right. Oh, I wasn't casting any aspersions.
Well, thank you so much for our chat today, Margie, and hopefully people have learnt a lot and just opened their mind a bit on what is available for looking at health on a very deep level. So, yeah, it's been a wonderful chat.
Thanks for inviting me along. Thank you.
Pleasure. See you later.
See you.
If you loved or learnt something in this episode, share it, and don't forget to subscribe. I look forward to bringing you next week's Awakening Conversation. Have a wonderful week.